Okay, so today we are going to continue with photosynthesis. Our last lecture we discussed redox reactions and the light reactions part of photosynthesis. So today we are going to move on to the rest of photosynthesis. So looking at photosynthesis in the two different groupings of reactions, the first one we looked at in our last lecture, the light reactions. So let's just summarize that and kind of recap. So light reactions always take place in the light. They capture solar energy. Water is split because remember we are donating electrons from water to our photosystem II complex, and in that process it releases oxygen. ATP and NADPH are produced in the light reactions. And so now we're going to move into the Calvin cycle, where we said that ATP and NADPH are going to be used. So the Calvin cycle can take place in the light or in the dark, it doesn't matter. Carbon dioxide is absorbed during this process, and it's reduced to create a carbohydrate. ATP and NADPH are used from the light reaction that we talked about a minute ago. And once that's used, then ADP and phosphorus, as well as NAD plus, are produced. And those are sent back to the light reaction to continue the process. So now let's delve into the Calvin cycle, since we've already talked a lot about the light reactions. Okay, so the Calvin cycle is broken down into three different parts. The first is carbon dioxide fixation. Sometimes it's also called carbon fixation. And what we mean by fixation is, taking basically the carbon that's in an inorganic form, and by linking carbon to another carbon, we are going to put it into an organic form that we can use. So fixation isn't specific to photosynthesis, it's not specific to carbon, but it is taking something from an inorganic form and putting it into a usable organic form. So the first step is carbon dioxide fixation. The second step is carbon dioxide reduction, and you'll remember what reduction means. And then the third step is the regeneration of what we call RUBP, or ribulose 1,5-bisphosphate is what it's actually called. So that's just a 5-carbon molecule. Keeping in mind that the Calvin cycle, unlike the light reactions that took place on the thylakoid membranes, the Calvin cycle takes place within the stroma of the chloroplast. So we're in a different location this time, so keep that in mind. So let's start with the fixation of carbon dioxide. So our reaction begins with carbon dioxide that has been absorbed from the atmosphere through the stomata and into our mesophyll cells, and keep in mind it's diffused in through the cells into the chloroplast, and now is in the stroma of our chloroplast. So we have our carbon dioxide, and actually carbon dioxide is going to combine with a 5-carbon molecule, and that is our ribulose 1,5-bisphosphate, or RUBP. And the enzyme that catalyzes this reaction is either called RUBP carboxylase, or what most people call it for short is Rubisco. And so here on the right, it shows you RUBP and carbon dioxide binding to Rubisco at its active site. Then we have our intermediate enzyme substrate complex, and then it during this process is this step right here, and that is where we get a 6-carbon chain, which is then cleaved into 2 pieces, and we wind up with 3-phosphoglycerate, or 3-PGA, and we have 2 of the 3 PGAs. So for every cycle that goes through using Rubisco, we wind up with 2 of our 3 PGA molecules. So that's the first stage of our Calvin cycle, is the fixation of carbon dioxide. Okay, so our second step in the Calvin cycle is carbon dioxide reduction. So remember, in the first step we took that carbon dioxide and fixed it, meaning that we attached it to other carbons. So I guess technically this could be just a carbon reduction reaction. And reduction, remember, means that we are gaining electrons or gaining hydrogen ions, usually both. So we start with what we ended with in the last slide, which is the 3 PGA molecule, and we have 6 of them at this point. And so we're going to phosphorylate that by using up an ATP, and so our intermediary step is a 1,3-biphosphoglycerate, and so you'll note here that we have 2 phosphate groups on that. And this ATP came from our light reaction. The next step then uses up the NADPH. And so this is where the reduction happens. So we donate the hydrogen ion and some electrons, and it releases a phosphate group. So then we are left with G3P, glyceraldehyde 3-phosphate, right here. So that is the reduced form of carbon. And G3P is important because it has more electrons and has higher energy and can be used to make a lot of things, including glucose. Okay, so then we're going to move into our third phase of the Calvin cycle, which is the regeneration of the ribulose 1,5-bisphosphate, which is the RUBP. And remember, that's that 5-carbon molecule that we started with when we said we were going to fix carbon dioxide. So we need to regenerate that. The nice thing about RUBP is that it's used through the Calvin cycle and then regenerated. So we don't, unless it gets worn out, we don't have to keep making new molecules of it. So in order to regenerate it, we're going to start where we left off with our reduction, which is our molecules of G3P. And so we had 6 of them produced. One of them is going to be used to make glucose. And the other 5 are going to be used to regenerate this RUBP. So note that we have 5 of them, and we have 1, 2, and 3 carbons for our G3P. And this is a process that takes energy. In fact, it's going to take 3 ATP molecules. And so we use up that energy to synthesize RUBP. And note that RUBP has 1, 2, 3, 4, 5 carbons. So we go from having 5 molecules with 3 carbons to 3 molecules with 5 carbons. So notice that we've kept the same number of carbons. And then, of course, we have added a phosphate group from the ATP. And so there we have the regeneration of our RUBP. And so we can start the cycle all over again. So this cycle has accomplished that we've created basically a monomer that can be used to make glucose, which is extremely important for all living organisms. And we've recycled one of the reactants that we need to even start this reaction. So our end product of the Calvin cycle is G3P, and so we did mention that it is mostly used to produce glucose, but it can be used to produce all kinds of things throughout the plant. So glucose would be the main molecule that we want to produce because plants and animals metabolize that for energy. So basically it stores a lot of energy. So the plant makes enough for itself. It usually makes enough to store. And then, of course, animals eating those plants also benefit from that as well. So we can produce G3P, and it can go through and make glucose phosphate, which can then combine with fructose phosphate to make sucrose. And sucrose is how plants transport sugars throughout its body. It can also produce starch. Starch is just a storage of energy basically for plants, kind of like we store fat. Plants store it in terms of starch. And then it's also used to make fatty acids and to make amino acids, so we can make oils and proteins. And it also is one of the beginning reactants to make cellulose, which we know is part of the cell wall of every plant cell. So the Calvin cycle is an extremely important cycle for all kinds of things throughout the plant. So when you think photosynthesis, we tend to think we make sugar and we make oxygen and we're done. And that is true, but it's a base reaction that has a domino effect throughout the plant, and not only for the plant, but for those of us who are consumers or heterotrophs of all of our producers. So here we just have photosynthesis written out as far as steps. I figured I'd give that to you for those who tend to be able to process a little better in word form, so that is there for you. So here is a way of looking at the Calvin cycle, keeping in mind that it is taking place in the stroma of the chloroplasts. So we have our carbon fixation, our carbon reduction, and then the regeneration of our UBP. And this is definitely a cycle that you will need to know. Notice the places where ATP is used and where NADPH is used. And then if you need to know what the different names of all the different carbon chains are, they are right here to make that easy for you. And then here is just a very simple summary of photosynthesis. We have our light reaction on the left, and that is when we collect the energy from the sun. Remember that we do split water in order to replenish the photosystems with electrons, and therefore there is a release of oxygen, and all of that takes place on the thylakoid membranes. And there is a production of ATP and NADPH from the light reactions. And those then get used in the Calvin cycle, which is in the stroma of the chloroplast. And again, we're using carbon dioxide in here. And through a series of steps, we use up ATP and produce ADP. We use up the NADPH and release NAD+, and we produce sugars. So that's the simplified version of photosynthesis, and now you know all the details. So everything we've talked about so far is what happens in most plants under ideal conditions. But what happens when things get too hot and too dry? If you'll remember, our plants, particularly on the leaves, have what we call stomata, and that's a hole in the leaves guarded by these guard cells. That's not important for you at the moment. But that is where carbon dioxide comes into the leaves, and that's what we need for going through the process of photosynthesis. However, there is an exchange for when you get carbon dioxide molecules in, there's an exchange of water molecules being released, also oxygen as well. So when it's very hot and dry, plants need to make sure that they don't lose too much water, and they close the stomata. And that's just a way of saving water, because remember, plants don't have skeleton. They count on the turgor pressure within each cell to hold the structure of the plant. So without that water, the plants would wilt, possibly die. So they close off those stomata when it's super dry. But what that means is that you're going to have a lack of carbon dioxide coming in. And in our typical plant, which by the way, we call those C3 plants. I won't go into why it's called C3, but most of the plants that we know in our typical plants are C3 plants. Well, in C3 plants, we use the enzyme Rubisco, remember, at the beginning in order to fix carbon. So the interesting thing about the Rubisco enzyme is that it can bind carbon dioxide, which is what it normally does in our photosynthesis process, but that active site can also bind oxygen. So when we decrease the amount of carbon dioxide that's available, that's less carbon dioxide to bind Rubisco. And at the same time, we are still producing oxygen because if it's still light outside, the light reaction is still happening. And remember, we split water to allow more electrons to replenish the photosystems. And when we do that, we're breaking apart water and releasing oxygen. So the oxygen concentration is increasing while the carbon dioxide concentration is not. So Rubisco will bind oxygen, and when it does, it goes through a process that we call photorespiration. And during this process, we produce one molecule of carbon, and that carbon, of course, is in the form of 3Pg. Normally, remember, we would be producing 2, and this just produces 1. So we've already decreased the amount of 3Pg being produced, and we also produce carbon dioxide. So that is photorespiration. It's not nearly as efficient as our photosynthesis for allowing all those reactions to occur. And so that's not ideal. So our normal plants then don't do very well when they're in those hot and dry conditions. However, there are other plants that do things a little differently because they are adapted for those environments. So I'm just going to briefly talk about those. Okay, so C4 plants have an adaptation for their photosynthetic reactions that partition out those processes by space. So first, let's take a look here in the bottom left at a cross-section of a typical leaf. So our C3 plants that we've been talking about tend to have chloroplasts all throughout their mesophyll cells. So we have palisade mesophyll cells here that line up just under the epidermis, and then we have our spongy mesophyll cells that we said have lots of airspace so that carbon dioxide can come through. And then what we have throughout a plant is what we call a vein, and that's our vascular tissue in plants. A little different than the vascular tissue in us, for instance. We have veins that carry blood, but it's a similar concept because our blood carries sugar to the places that need it. In plants, vascular tissue carries sugar and water to the various parts of the plant. If you think about a celery stick, you break it apart and peel it, those strings that are attached, those are the veins of a plant. And so the vein within the leaves and pretty much throughout the plant is covered by what we call a bundle sheath. So this row of cells right here that encapsulates the vein, that's called the bundle sheath. And in our typical C3 plant, those cells do not contain chloroplasts. Well, when we shift and look at a C4 plant, the mesophyll cells are arranged differently. They form a concentric ring around the bundle sheath cells. And notice that the bundle sheath cells themselves also this time contain chloroplast. So things are a little bit different as well in C4 plants in that it uses an enzyme called PEP carboxylase to fix carbon to a compound called oxaloacetate. Oxaloacetate is then reduced to a form called malate. And all of that takes place in those mesophyll cells. Now, if we then take malate and pump it into the bundle sheath cells in a different location, we can break down malate and it produces in order to form carbon dioxide. And that carbon dioxide then can bind rubisco. And that's where the rest of that Calvin cycle will take place, because we have a ready supply of carbon dioxide so that oxygen doesn't bind rubisco and we don't have to have photorespiration. So this allows C4 plants to thrive well in really hot conditions. So if you think about here in North Carolina, think mid-July, super hot, super dry. Don't we almost always have a drought? And if you look at some of those neighborhoods where we have fescue lawns, that lovely lush green grass that in the spring is beautiful. That fescue grass is a C3 plant. It thrives great when conditions are plenty of water and cooler conditions. But as we get into July around here, the fescue struggles. And what comes in place of that is things like crabgrass, which I have pictured here, which is a weed. A lot of our weeds are C4 plants that will thrive really well in super hot, super dry conditions. And that's because of this partitioning by space, doing the parts of photosynthesis in two different cell types. So another type of plant is called a CAM plant. And these are typically your succulent plants, which tend to be a fad right now of decoration. But the succulent plants are the ones that have real thick leaves. They're full of water. They tend to be smaller plants. So the CAM plant gets around the photorespiration problem by partitioning its photosynthesis in time. It will separate out when it does certain reactions. So at night, it can convert carbon dioxide, it can fix carbon dioxide to oxaloacetate, again reducing it to malate, similar to the C4 plants. But then it takes the Calvin cycle and rubisco into the daytime. So there's enough carbon dioxide to bind to the rubisco because of the reduced malate and other organic molecules that can be broken down and release carbon dioxide. And then as the Calvin cycle is happening during the day, that's so that it can utilize the NADPH and the ATP that's produced from the light reactions. But it already has enough carbon dioxide that we don't have to worry about oxygen binding all the rubisco. So partitioning out our pathways via time ensures that no photorespiration occurs. So plants have all kinds of different adaptations based on where they live. And remember, we said that was a characteristics of a living thing, was the ability to adapt. So I hope that you get a sense of awe when you look at all the details in such a tiny process that sustains life on Earth. And now I hope you will look at plants a little differently when you see them. Just the amazing things that are occurring in just one cell of a plant is mind-blowing. So anyway, hope you get an appreciation for that, and our next lecture we're going to start talking about cellular respiration, so how we can use these carbohydrates that are produced during photosynthesis.